virtually to be able to tell you a little bit about some of the work that we've been doing in polymerization-induced self-assembly over the last couple of years. So I just thought I'd maybe start with a brief overview of the kind of research that we're interested in my group. I think one of the unifying themes that we're interested in trying to work towards is thinking about how we can precisely control chemistry across the length scales. So if, just get the pointer. So if we think we do work on non-invasive probes, so we're interested in developing these bromo and chloromaleimide structures, which are really bright self-reporting probes, and we've developed those as a tool to help us with in understanding the processes in some of these longer length scale materials. We've also got an interest in DNA templated synthesis and DNA nanotechnology as a method for programmable synthesis, and then moving through the length scales, thinking about how we're interested in confinement as a way to be able to control the chemical environment of things such as catalysts and these sort of bio-inspired nanoreactors. We've also in recent years become increasingly interested in anisotropic constructs, specifically rod and platelet-like structures, and thinking about how we could create these in a precisely controlled way. We also are interested in how we can kind of bring some of these polymeric structures together to actually allow for responsive materials, and this is a work that we do actually closely in collaboration with industry to look at how we can create these functional materials for particular applications. So self-assembly is something that kind of underpins all the work that we do in the group primarily, and there's kind of two main elements of self-assembly that we're most interested in and have been in recent years. The first is crystallization-driven self-assembly, where we use a semi-crystalline block polymer, and we nucleate and then seed to create a seed which we can, in a very controlled manner, grow these self-assembled constructs. And just shown here are some of the diamond platelets that we were able to make, where we can really quite carefully control both the dispersity and size of these constructs. But today I'd like to tell you a little bit more about the work that we've been doing on polymerization-induced self-assembly, or PISA, and we've been doing this both from using both raft polymerizations, but also rump polymerizations. And just highlighted here, as I said, is a review actually that was written by a fantastic PhD student in the group who's just recently graduated, highlighting kind of the field of rump PISA in recent years. So just to kind of outline polymerization-induced self-assembly in case people aren't necessarily familiar with it, very much the concept is that you start with a soluble macro CTA and you polymerize a miscible monomer. That miscible monomer, as you polymerize it, it goes from hydrophilic to hydrophobic, and then during the polymerization process you get this self-assembly occurs, hence the name polymerization-induced self-assembly, and you create in the same way you would in just a conventional polymerization process, where you form an amphiphile with a hydrophilic and hydrophobic blocks, and those block ratios and packing parameters determine the morphology of the construct that you obtain, be that spheres, worms, or vesicles. But PISA has some interesting advantages in that it's extremely simple. So you do the polymerization and assembly in one step. It can environmentally friendly in the sense that you could use, often done in aqueous media. Very scalable, because you'll see they're often done relatively high weight solids, and it's very easy to access higher order morphologies, so interesting different shapes of nanostructures. So just to highlight some of the really nice work that's been done in this area, as I said, Steve Arms has done some really lovely work where in this case he takes a PEG macro CTA and then through raft polymerizes HPMA to form this block, and through this polymerization-induced self-assembly process you can see here the range of morphologies that he can produce. And then more recently there's been some really nice work from Summerlin, Tan and Zhang, who showed that actually you could use aqueous photopisa, so we're now starting to be able to use very mild polymerization conditions, in this case just 405 nanometer light, to be able to enable this polymerization and then indeed create similarly complex morphologies. And as I said, the high weight solids content is one of the key advantages, and the single step, and as I said, especially with photo-initiated raft, really mild an aqueous reaction, mild aqueous reaction conditions, and you can very predictably by forming a phase diagram be able to predict the morphologies that are formed within your system, and given that the polymerizations go to very high conversion, often no purification is required. And just to highlight, there was a very nice, what's now a few years old, a recent review on light-mediated PISA from Cyril Boyer that is pretty quite extensive and may be of interest. So some of the early work that we did a number of years ago was looking at the comparison of photo and thermal initiated PISA. And one of the things that we noted, so if you just look at the plot here, if you look, the photo initiated PISA is significantly faster with, and a characteristic feature of PISA polymerizations is that you see this acceleration in rate of polymerization upon assembly. So as I said, as the construct forms, you then get this step change and increase in polymerization rate. And you'll see for the photo system, for the same polymerization initiated in two different ways, either thermally or photo, you can see that the point at which this assembly occurs is around 15 minutes for the photo and about 200 minutes for the thermal. And the result of that is, if you look at just the two phase diagrams, this is the photo and the thermal, you actually have, as I said, hopefully can see quite quickly that the photo initiated PISA leads to higher order structures when compared to, as I said, thermal. But you also see quite a significant loss of end groups. And this is one of the first PISA bits of work we did in the group, and it was really started by Lewis Blackman, who is a PhD student who's now at CSIRO. But one of the things PISA is also interesting for is for encapsulation. So as you imagine, if you start with, say, for just example, a commercially available polymer such as a PEG, you functionize it with whatever, say, initiating group you want to use for your to form your second block. You can purify it, and then in one step you can perform the polymerization in the presence of something that you'd like to encapsulate, in this case just shown as an enzyme, and potentially use photo initiated systems, which, as I said, very mild, so it doesn't damage delicate enzymes, etc. You can then do the chain extension or self-assemble in one pot, and then just purify to be able to access encapsulated species within the constructs that you're making, in this case targeting vesicles. And this has been used really effectively by Steve Arms. He showed really elegantly how you could encapsulate silicon nanoparticles. And then also from Summerlin and Tan and Zhang again, they were able to show very nicely that they could encapsulate BSA. And we've been interested for a number of years in trying to encapsulate active enzymes that might allow for us to be able to scope out the function, the protection, but also functionality of such enzymes within our polymeric constructs. So the concept being that if you imagine we take a PEG macro CTA, which has a raft end group, we then polymerize just HPMA. It commercially comes as two different isomers, but we just polymerize the mixture, commercial mixture. You can imagine doing this polymerization under mild aqueous PISA conditions, say with photo initiation, with a functional protein, and then with your protein embedded within your construct, it's protected from antibodies and proteases, but the size selective permeability of the membrane allows for reactants to be able to permeate and access the enzyme and products to diffuse out, so you can still retain the functionality of the enzyme when embedded within the construct. And we demonstrated this for two enzymes, HRP and GOX. We looked at loading these, performing our conventional PISA polymerizations in the presence of these two enzymes. The size and properties of the polymerisomes formed were very similar to that which was formed without the presence of the enzymes. We get loading efficiencies of, as you see, around 28-24%, and for the two enzymes, actually very reproducible. And we were able to show that when say for example the HRP in this case, using standard kind of assay, colorimetric assays for these enzymes, we could show quite readily that the enzymes were still very active and retained a good portion of their activity relative to the free enzymes. And so, as I said, this demonstrated the mildness of the polymerization conditions, but also the size selective permeability of the membrane. We also demonstrated that this could actually be used in a cascade reaction, so GOX and HRP undergo this cascade reaction with glucose, leading to this oxidized dimer and the production of this colorimetric reaction. And only when these two enzymes, or these two polymerisomes, which had the two different encapsulated enzymes within them, were present, were we able to see the expected response. So we were able to see that we could readily compartmentalize these enzymes and allow for this cascade reaction between the two polymerisomes with around, as I said, close to about 50% of activity retention. One of the things we were really quite interested in was whether we could actually use this size-selected permeability of the membrane to allow for enhanced therapeutic function. So one of the things that we were interested in was looking at this therapeutic protein, asparaginase, or ASNS, and this is a treatment that is used as injected treatment for acute lymphatic leukemia, and it functions by depleting the L-asparagine and prevents tumor growth. So you can hopefully see the asparaginase converts the L-asparagine to L-aspartate and Kidrolase is the drug on the market. It can also be pegylated and is pegylated to form a new drug called Oncaspar. There are some immune response issues and hypersensitivity, and there are some drug incompatible issues with this therapeutic protein. So we wanted to see if we could utilize this therapeutic protein and embed it within our construct to allow for improved activity. So if we think of what we're proposing doing is taking our ASNS, which is a therapeutic protein, using our PEG macro-CTA, our standard HPMS monomer, doing our photo RAF polymerization to encapsulate the ASNS within the construct. And the way that we propose that this would work, so as I've said, the asparagine is converted to aspartate by this ASNS. And in healthy cells, there's a normal level of ASNS, and the addition of ANS as a therapeutic has very little effect. However, cancer cells cannot have very low levels of ASNS, and they cannot make asparagine, which is a non-essential amino acid. So it has to recruit asparagine from the blood, and if we have our therapeutic enzyme delivered, as I said, then we see a reduction in asparagine, so that the ASNS has a large effect on cancer cells because it becomes starved of the asparagine, and hence does not, as I said, allows for starvation of the cancer cells and selective cancer cell death rather than healthy cell death. And this is the same mode of action that's used in the clinic, but just with straight asparagine. We wanted to use this, as I said, within our polymer nanoconstructs. So again, our hypothesis is that we have our therapeutic protein embedded within our polymer micelle. It will be protected from proteases, protected from antibodies, but would still be able to be size selective to allow for the asparagine to permeate in and react with the ASNS, and also allow for the aspartate, which is produced, to be removed. If we just look at very briefly at the TM, we form nice, similar polymerosomes to which we form without the presence of the therapeutic enzyme. We see relatively narrow dispersity, as I said, very much as we would expect. And the idea was to really try and see if protection of the therapeutic protein within our polymerosome would improve the therapeutic activity. So here's some of the data that we were able to obtain. So if we just look at the native ASNS and look at the binding of the antibody, we see high binding when we just have the free therapeutic protein. However, when pegylated, which, as I said, is one of the other modes of treatment, you see there is a reduction in binding, and indeed when we load our therapeutic within the polymerosome, we see a complete reduction, and there's no interaction or binding at all with the antibody, which is very promising. We also, one of the other challenges with therapeutic proteins is very much the things such as chymotrypsin, which can act as a proteases, which can destroy and act on the enzyme, on the protein, and affect activity. And if you look at just the free therapeutic, when incubated with alpha chymotrypsin for a couple of hours, we see a complete loss of activity. The pegylated actually offers very little protection against proteases, or encapsulation offers really significantly improved resilience against proteases, and this is simply because of the size selective barrier. And just shown here is this a similar experiment, but now this is performed over 7 days. So we really have a very good long life and good stability of this using this our encapsulated approach. So we've been able to increase the proteolytic stability and reduce the antibody recognition, which is great. And this in turn we propose would reduce the risk of an immune response. But we really wanted to see if we could, in vitro, demonstrate the efficacy of these therapeutic nanoreactors. So we tried to mimic the depletion of asparagine in the surrounding environment, which is what happens as I said, in a leukemia. So the way we did this was to assess the ability of our polymerasomes to inhibit cell proliferation in vitro. So we generated cells that were deficient in our ASNS to simulate leukemia, and then A549 cells were gene silenced with the appropriate siRNA, making them dependent on an external asparagine to proliferate. So we took our ASNS vesicles in growth media to provide us with a depleted environment, centrifuged to remove the vesicles, and then took the media and used this supernatant as our reduced asparagine environment, and then used that treated growth media with our gene silent cells and with the normal growth media. And after 7 days, we can see that just with the normal media and proliferation and just the control with empty vesicles, we see no change in proliferation. But we were able to get about a 50% drop in proliferation when we were using our ASNS vesicles. And this really does highlight that the encapsulation of this therapeutic enzyme within these polymerasomes still allows for increased activity. We propose it would also allow for increased circulation times due to the larger size, and this coating effect, which is within the polymerasome, is much more effective than just a pegylation, because it really does protect the delicate and active enzyme from or therapeutic from other things such as the antibodies and proteases. So we've done all of this with RAFT polymerization. We also were interested in moving to some other polymerization mechanisms to allow to try and explore a bit more the versatility of the PISA process. So we became interested in using ROMP PISA, so using these norbornene type monomers. One of the advantages with ROMP is very much that it's very tolerant to a variety of functional groups, and also in air, so really very fast as well. And we proposed that we could think about taking, if you imagine we have our solvent soluble macroinitiator, again using the same principle of having a core forming block and then being able to make these nano objects. We were very keen to do this in aqueous media, which is actually quite challenging. Doing ROMP in water is rather difficult. You either need to use either a poorly defined catalyst, or it can be very difficult to get good polymerization control, and you really do see often quite poor polymerization control. So one of the first things we needed to do was to try and think about a method that may allow us to more readily be able to perform these ROMP PISA reactions in aqueous media without the need for complicated catalyst synthesis. So what we looked at was what we were terming our macroinitiator approach. So just a very simple concept where we just take a commercially available Grubbs type catalyst. We just add on a few water miscible monomer units onto the catalyst, and this then makes it now soluble. So rather than the conventional is to modify the ligands to have paired groups to solubilize the catalyst, we just took an insoluble catalyst and added a few monomer units to make this water water-soluble macroinitiator, that then this renders the catalyst water-soluble, and then we can use that water-soluble catalyst and polymerize any monomer that we want to be able to then form the basis of our hydrophilic macro-CTA and the first block in our PISA polymerizations. And one of the things we were quite interested in, and we become interested through a collaboration with Rob Mathers from UPenn, was actually thinking about the choice of monomer that you use for PISA. So as I said earlier, the concept of PISA is that you start with a water, if you're doing an aqueous PISA, you start with a water-soluble, you start with a water-soluble macro-CTA, and then you polymerize a monomer that is water-miscible. In the early stages of that second block formation, the very short DPs, that polymer is water-soluble, but then it undergoes a transition to become not water-soluble and transitions from hydrophilic to hydrophobic, and that leads to the assembly. So that growth of polymer during polymerization and that transition from hydrophilic to hydrophobic is really key in the PISA mechanism, but it's actually very difficult to know which monomers might do that. So a number of years ago, we, working with Rob Mathers, we looked at applying a very simple log P over surface area calculation, which allows us to be able to probe how the water solubility of the polymer changes as the polymer grows. And just shown here is the example that we had for the ROMP PISA system. We've also done that. Our initial papers were in RAF PISA, but we also were able to apply it very successfully for ROMP PISA, so that we can actually draw a monomer that we might want to make, look at it how as we grow. As you see, these are relatively small oligomers, but we can see how we can determine the hydrophilicity or hydrophobicity and the increase in hydrophilicity or hydrophobicity as the polymer grows, to allow us to, rather than make the monomer and try to see if it works, we can get a good sense of whether we think it might work based on the change in hydrophobicity or hydrophilicity as the chain grows. And we showed this last year for ROMP PISA, and this is what we've been using to be able to identify monomers that will undergo ROMP PISA. And we've also been able to use it quite effectively to be able to compare the different morphologies that are formed within the same system, within different systems, but with the same degree of polymerization. So just the example shown here, if we think of our range of different norbornene monomers that we have either an ester, an amide, or an imide that are the functional groups that are the linker group that attach, say, our alcohol functionality in this case to the norbornene monomer. And the change in those linker chemistries has quite dramatic changes in the resultant morphology in PISA. So these are all have a degree of polymerization of 120, and you can see the imides form spheres, the imides form spheres, the amides form worms, and the esters form vesicles. So again, the chemistry, that linker chemistry is really important in as I said, in the resultant morphology. We can show that again, as I said, and again, if you look at the dependent functional group and how that affects the morphology formed, we see a very similar trend again, where we get higher order morphologies as we increase polymer hydrophobicity. So this helps us kind of plan our phase diagrams and plan some of our targeted DPs, etc., a little bit better. So one of the interesting features of ROMP PISA was that these these imide-based polynorbornenes have quite a rod-like structure. So this is independent of the side chain chemistry, but they also have extremely fast propagation kinetics. So if we just use the conventional pegylated G3, which is the conventional approach for aqueous ROMP, with our macro-initiated approach, we're now seeing very fast kinetics, and this actually leads to the evolution of quite unique morphologies. So if we just very briefly compare the RAF PISA phase diagram with the ROMP PISA phase diagram, if you look in the RAF PISA, we really only see vesicles or polymerosomes at high concentration, high DP. If you look at the ROMP PISA, you'll see that the vesicle window is much larger. We see higher order structures, and we see and we propose this is in part due to the rod-like nature of the polynorbornenes, and hence when packing within assembled structures disfavor higher curvature structures such as spherical or worm-like micelles and favors polymerosomes. But this, so as I said, we've seen vesicles form over a broad range of DPs, which, as I said, we were quite interested in exploring how energy is built up and released in these systems. And actually, if we move to higher DPs, what might happen? Do we get vesicle fusion or rearrangement? And we hypothesized that these might be kinetically trapped morphologies with high membrane tensions as a result of the really high propagation rate relative to self-assembly. So we're getting these kinetically trapped morphologies. As I said, we want to see if we could explore this in a little bit more detail. So this was the experimental design where we just take our G3 Grubbs catalyst, and we then add our water-soluble 10 units of this water-soluble macro initiator, which is the same in both, is different in both in the two different polymerizations that we're looking at. This is our water-soluble polymer, and we have, sorry, our piezo-active monomer. We've done this reaction with two different systems, one which has a PEG pendant group and one with an amine, to try and explore how the chronochemistry and core block length influence the morphology. And we're looking at DPs beyond 60 because this is where we think we see interesting morphological development. And the way that we analysed this, because we were quite interested in having done some initial work, we saw that we were seeing some non-spherical constructs. So we did some image day particle analysis, and we defined the circularity of the complex, which is basically the cylinder versus sphere, which is decided using this formula for particle circularity. If the C is equal to 1, we have a spherical particle. If the C is less than about 0.7, this means we have an elongated particle. I'm not going to dwell on mass, but just important to highlight the C value was something that we were using. So if we just look at the system where we have our aiming block, you can see from the SEC traces and data, we have a well-controlled polymerization producing relatively narrow dispersity polymers. If we run through the DPs, increasing DP from 120 through to 420, you can see the particles increase in size from about 40 nanometers to about 80 nanometers. The circularity stays relatively similar, about 0.79, 0.75, 0.74. They're very similar. You can see these start to look a little bit elongated. So we're just seeing spherical constructs throughout, and vesicles throughout the whole range of, as I said, even at really rather high DPs. Interestingly, for the PEG-based system, we observe something different. So if we start here at a DP 60, and then at DP 120, we still have particles of increase in size. We still have a circularity index that suggests they're still relatively spherical. That starts to decrease quite dramatically, and hopefully you can see clearly from the TEM images that we start to form these worm-like structures, and we term these tubosomes. So these now are these elongated long structures that have formed from the original polymerosomes for this PEG-based system. So if we look at a little bit more detail at these images, perhaps you can see you can look at some fused vesicles, some single vesicles, and vesicles that have undergone multiple fusion events, and also some potentially fusion events. So one of the things we were interested in, strong indication that we're getting fusion. Well, when was fusion happening? Was it happening post polymerization or during polymerization? So one of the things we to explore, and we checked that the fusion basically requires polymerization to occur. So if you look, this is just for our PEG system, as I said, as the DP 120 polymers as prepared, if we just leave them and age them for a month, they look exactly the same. So the constructs we're making are stable and don't undergo a morphology transition over time. If we take, again, and that's whether it's the starting spherical polymerosome or our tubosomes, with after a month of aging, they're indeed very similar. So they're very similar constructs. So if we think about what might be happening, this is a soft matter paper from a number of years ago from another group that describes dissipative particle dynamics, and the simulations that they show that fusion basically occurs between vesicles simultaneously if the vesicles can adhere to one another and maintain in close contact, and if there's sufficient membrane tension to overcome the energy barriers of fusion. And they showed some really nice work on this around the tension required for that. And the fusion helps release high membrane tension. So one of the things we thought was, well, if you remember for the amine system, we didn't see any polymerisome fusion. We just saw, I said, they were retained to spherical constructs. So we proposed that with the amine-based system, we were not able to get particles adhering to each other and not being able to maintain close contact, so, because of the charged nature of the amine. So we thought if we shielded those amine interactions, would we be able to see fusion? And the answer is yes. So just doing the same polymerization I showed you a couple of slides ago where we hadn't seen a change in morphology, now you can see quite clearly when we do this, repeat this in a salty solution which screens charges, you can now see we're seeing very similar fusion events and the formation of these tubosomes as we saw for the PEG-based system. And this does again confirm that close contact is required to enable, as you might expect, such fusion processes. But just close contact's not enough. We actually need to think about the vesicle energetics and actually the tension that we're building up as we in our systems, and why our systems may want to fuse. And again, this may be because, as if you remember, I said that we have an extremely fast polymerization. So you can imagine that our vesicle membranes are sort of becoming charged with high tension as the length of their polymers increases. As I said, and this to try to compensate for the static curvature and surface area, so we're getting this build-up of tension, as I said. And we propose that this build-up, alleviation of this tension, is the driving force for vesicle fusion. As I said, it reduces the energy of bending of the system. So how might energy be minimized during our rump PISA? If we start with a very low tension and with our initial polymerisome, as we polymerize, that tension starts to build as we polymerize within that membrane. So we start to reach our higher tension system. We can either get, if the polymerization is rather slow, you could imagine how you would then get rearrangements such as unimer exchange that would then lead to either, as I said, a loss of construct and reduction in energy. The other way to relieve the tension is to think about vesicle fusion, and we get high energy for vesicle fusion, but then actually reduction in energy as we form these low tension tubosomes and the worm-like type structures that we've seen in our system. And this fusion process is a step growth-like process, which we would expect if we're, as I said, as the growth process we might expect. So we can fit, as we might expect, to the Carruthers equation and look at, and using just dimensions from TEM, we can be able to see, hopefully see quite clearly that we can look at the LW over LN of our systems and be able to theoretically predict the values we might expect, and it matches very well with the theory, indicating that we do indeed have this step growth-like process and growth of tubosomes. So as I said, our proposed mechanism is that we have these low tension polymerisomes. The polymerization proceeds very quickly and builds up tension quite quickly, so we have a very high energy of bending, and fusion helps release that. But then as polymerization continues, we build up the tension again and high energy of bending, and again a cycle of building and releasing tension allows for a growth of our tubosomes, and these tubosomes that we form are stable, and fusion stops after the polymerization stops. So we're able to form these tubosome type structures, as I said, but we were able to show, I don't have time to tell you today, but we were able to show some work, and one of the interesting elements of this is that it allows for lumen-lumen mixing. So we've been able to show that using a variety of dyes, etc., to be able to confirm, as I said, this fusion type mechanism. It just remains me to thank Team PISA, I guess. So Lewis Blackman, as I said, was the first PhD student in the group. He graduated a couple of years ago, and as I said, is now at CSIRO. He did a lot of work at the RAFT in PISA. Then Spiros Varlas was the PhD student who took over. He graduated in the last couple of months and has just started a position, a postdoctoral position at UCL. Ably assisted by Jeff Foster, who's fantastic postdoc and group leader in my team. He's just about to start a position in Sandia National Labs in the US. Fantastic two PhD students, Rob, who then went on to stay to postdoc, and Yuji, who's soon to finish her PhD. I should acknowledge our collaborators on the ASNS work was with Matt Gibson at the University of Warwick, and the LogP work with Rob Mathers at the University of Pennsylvania. And then, of course, just thank the rest of my group and thank various funding bodies, specifically at the University of Birmingham, for funding the EPSRC and the ERC. And thank you all very much for your attention. I'm not sure if there's time for questions, but